All right, in this video, I'll continue the conversation about autonomic nervous system, the two divisions, the sympathetic and the parasympathetic. I showed in the last video that the parasympathetic and the somatic is not part of the autonomic. The somatic is voluntary, but they both use acetylcholine at the effector organs. So here in the postganglionic, postsynaptic membrane of these organs, as well as the postsynaptic membrane, really the motor end plate of the skeletal muscle right here. They all use acetylcholine. Now, I would like you to spend some time, read the section of a textbook titled Exploring the Concept of Receptor Subtypes in chapter 15. And particularly from the subheader titled What is a Receptor Subtype, all the way to the subheader titled Why Do Receptors Matter? I'd like you to post your summary of your response, how you understand this in your own words, one or two, either question 1 or question 2. This is really important to help you understand the next segment, which begins to talk about where and how different drugs act, and basically why are certain drugs selective and cause predictable effects through specific receptors and tissue types, and why others are not. This knowledge is important when you are prescribing medicines, because not all cholinergic agonists are created the same. There will be a time you would want a particular subtype versus another. So please do this and post that on the discussion board. Or print a copy and bring it to class. So where are we now? So this is table 15.3. One of the things about this table that people find confusing is now we are in, where is this happening? This is in the postsynaptic membrane in the effector organ. So this is all effector organs. Now, in the left column is the type of receptors, the subtypes. So it is shown here 3 different receptor subtypes. They're all cholinergic receptor subtypes. Let's get, so there's the nicotinic and the muscarinic. Under the nicotinic, there are the ones that are found in the neuron. Those are found in the ganglia. Remember, the ganglion is the point between the neuron that exits from the central nervous system and the neuron that goes on to the actual effector organ. So that ganglion, that acetylcholine actin there, acts on nicotinic neuronal or nicotinic N type. Okay. You also have this in the adrenal medulla that I will talk about later. And then there's the nicotinic M as in man or money, and nicotinic M is the neuromuscular, is the type of acetylcholine that you find in the neuromuscular junction in the somatic system. I actually like nicotinic money because, when you work, you make money. So voluntarily, nobody works involuntarily. So nicotinic muscular is what you find in the somatic nervous system. Again, not autonomic, somatic. So those 2 subtypes are actually different, NN and NM. Some drugs will bind one and not the other, as you will see as we go along. And then you have the muscarinic. Now, the muscarinic is important because this is what controls directly, so direct agonist or direct antagonist. These are the receptors on the postsynaptic effector organ that controls your involuntary responses. So if you think about every involuntary response you can have, your gut's beginning to growl around midday, That's muscles contracting. You're salivating around 1 p.m. when it's lunchtime. That's a gland releasing saliva into your mouth. Um, those salivary gland in your mouth, the smooth muscles in your stomach, and your GI tract in general, and so on, those are all effector organs that are controlled by the muscarinic. What is shown here that I'll just describe here. There's more on the table, and I would require that you understand the entire table. Is in the eye. So in the eye, the actual pupil itself is just the space that opens up to let light in or out. What controls the diameter of that pupil is actually your autonomic nervous system. So remember the pupillary reflex, you shine light into someone's eye, the pupil constricts to limit the amount of light that gets into the eye to prevent damage in the retina. Now, so when the eye constricts like that, we call that miosis, M-I-O-S-I-S. That is a cholinergic response. Acetylcholine binds to muscarinic receptors on the sphincter muscles. Okay, there are two different muscles that can control the radius of the diameter of the pupil. One is the sphincter. So the sphincter muscles are arranged in a concentric fashion around the pupil. That's what makes the pupil. So when they contract, the hole narrows, and so you have that. Okay, so that is the sphincter muscles, and it responds to acetylcholine binding to muscarinic receptors to cause that miosis. Okay. Now you also have radial muscles. So the radial muscles pull the pupil outwards when it contracts. So essentially, it opens up the eye. Okay, but that radial muscle is not controlled by muscarinic receptors. It's controlled by adrenergic receptors. Remember, that's under the sympathetic division. I have not covered that yet, but the point is, when you perceive danger, your eyes get all wide. That's because when you perceive danger, your body makes a lot of norepinephrine or epinephrine, fight or flight. And one of the things that norepinephrine does is it binds to adrenergic type 1, alpha type 1 receptors on the radial muscles. And when it binds to that, when those radial muscles contract, they pull open the pupil. So this here illustrates something I showed earlier when we looked at the dual innervation of the heart. In this case, the organ function is controlled by the adrenergic and the cholinergic. That is the eye's function. The pupillary diameter is controlled by both, but they are controlled by different muscles, receptors in different muscles. So in the cholinergic, it controls the sphincter. In the adrenergic, it controls the radial. But the point is, it's a yin-yang system. They have opposing effects. And that makes sense. You are not relaxed, rest and digest, and sleepy at the same time with your eyes wide awake. Same thing when you perceive danger, you will not, you need your pupils to be as wide as possible so you can have as much light to decide whether to fight or to flee. Okay, so next here is a diagram that shows the cholinergic terminal, and this could be nicotinic or muscarinic. Now you're familiar with those terms, and it shows how acetylcholine is made in the synaptic terminal, and acetylcholine is stored in the vesicle, and then it's released through synaptic release here, and acetylcholine, when it gets here, will bind to the receptor. If this tissue, this postganglionic cell, if it was a neuron, like you had in the ganglion, it will be nicotinic neuronal. If this postganglionic cell was a skeletal muscle, it will be nicotinic muscular. If this postganglionic cell was an involuntary muscle or gland, then it will be the muscarinic receptor. And then once it binds, you have this enzyme here, acetylcholinesterase, that breaks down acetylcholine, it's choline and acetate, and essentially inactivates. Please note, this is the predominant mechanism, the major mechanism by which acetylcholine's activity here is terminated. That's the only way. Even though you see choline being reuptaken, that reuptake is secondary to this breakdown, and this is what drives the termination. So finally, now you've learned about different functions in the cholinergic division. I'd like you to put on here some drugs that you think can be a direct or indirect agonist or antagonist at this terminal, neurotransmission terminal. All right, thank you.